So then we'd like to think, well, how can we avoid this? What are therapies that we could do or apply to a patient to shift this distribution of T cell states? So we want to go away from being in the terminal exhausted state and favor progenitor T cells in these effector T cell states that are good at dividing, making more of themselves, and killing tumors. So really, part of this challenge is thinking about how we can shift the distributions of these T cell states. And there are therapies that do exactly this. And the most exciting class of therapies are cancer immunotherapies. And cancer immunotherapies are therapies that actually just boost the immune system's ability to fight cancer. And there are two main therapies out there: immune checkpoint blockade and CAR T cell therapy. And both of these therapies act on T cells. So they're both modifying the T cells in some way to make them better at killing cancer. And so I like, looking at what oncologists have said about these immunotherapies and just seeing, how much improvement there's been since we've started using immunotherapies. So here's a quote from Jason Luke saying, we're talking about diseases where we used to tell patients during their first clinical visit that they were going to die from this cancer. So really, immunotherapies in certain cases have made a big difference. So there's two types of immunotherapies. We're first going to talk about immune checkpoint blockade. So let's return to a cartoon that's similar to what you've seen before, where we have a T cell engaging with a tumor cell or cancer cell. On the surface of the T cell is a T cell receptor that recognizes the non-self antigen from the cancer cell. So you saw this earlier. And actually on T cells, there's many molecules. So another molecule on the surface of the T cell, other than the T cell receptor, is PD-1. And PD-1 is a molecule that is a major regulator of T cell exhaustion. And what happens is tumor cells express another molecule, PD-L1, which binds to PD-1 and turns off the T cells and keeps them from activating their killing response. So anything that could really disrupt this interaction would free the T cells and make them better killers. So when this interaction is happening, the T cells are kind of being driven into this terminal exhausted state. And so the idea with checkpoint blockade is to disrupt or block this interaction by designing proteins or antibodies that bind to PD-1 and block the interaction from ever happening. So in checkpoint blockade, we have these antibodies. They block this interaction, and this frees up the T cell to not go into the terminal exhausted state and to stay in a progenitor state or an effector state and be able to kill the tumor cells. So now let's look at an example of one of the first clinical trials that was done using immune checkpoint blockade. So one of the first trials was ipilimumab, which is an advanced melanoma. So that's a very bad stage of the melanoma disease. And here we're looking at the results of that clinical trial data, where on the y-axis we have overall survival, and then on the x-axis we have how long the patients survived. And so here we're seeing the control arm of the trial, where these are patients who did not receive the checkpoint blockade. And then here we're seeing the treatment with ipilimumab. And so at first glance, this is a significant difference, but it's maybe not a huge difference. But what you're seeing really in this data is that, in the control arm of the trial, patients are, unfortunately dying, from 8 to 12 to 16 months. And with the ipilimumab treatment, you have patients living out to like 60 months, a certain subset of the patients. So not everybody responds to ipilimumab, but those who do actually live quite longer. And, this was a trial that was done in 2010. And since then, in the last 10 years, there's been a lot of advancements in immune checkpoint blockade. And I like this quote from Jed Walchuk at Memorial Sloan Kettering, saying that when he first started treating patients in the year 2000, the average survival for something like advanced melanoma was only 7 months. And now with the latest sort of FDA approved checkpoint inhibitors, average survival has increased to over 6 years. So things have really gotten better and better and better, but still we have this issue that not all patients respond to immune checkpoint blockade. So the other form of cancer immunotherapy, or the other main form, is CAR T cell therapy. And again, like checkpoint blockade, this is working with T cells. So CAR T cell therapy is really clever and cool. And how it works is it's actually a living drug that's derived from the patient. And so what happens is the cancer patient gets a blood draw, and scientists in the lab extract T cells from that blood draw, and then they genetically engineer the T cells to express on their surface another molecule called a chimeric antigen receptor, or CAR. And this CAR is engineered to actually recognize the non-self antigen on the surface of the cancer cell. And so this is something that really is, very clever. And then what happens is you take this CAR T cell that's now been engineered to recognize the cancer cell, you grow up tons and tons of them, millions of the cells, and you infuse them back into the patient. And then the CAR T cell goes from the blood and traffics to the tumor and starts to kill the tumor cells because it recognizes the non-self antigen on the surface of those tumor cells. So this is really amazing. Here's an example. It's been used to treat diffuse large B-cell lymphoma, and 50% of treated patients have a durable, complete response. However, again, what about the other 50% of the patients? So cancer immunotherapies are amazing in so many ways, and they really transformed the lives of a lot of cancer patients. However, the big problem everybody is really focused on is, why do these therapies only work for some people and for some cancer types? And can we really get these therapies to work for more individuals across more types of cancer? So what are some of the reasons cancer immunotherapy fails? So first, in immune checkpoint blockade, not all cancer cells are acting through PD-1 or the related molecule CTLA-4. So in these cases, checkpoint blockade just doesn't work because you don't have the appropriate target. Now, also in cancer, individuals can evolve resistance to the checkpoint blockade therapy. There are changes in the cancer that result in the cancer becoming resistant to checkpoint blockade. And this can either work by the cancer cells starting to directly inhibit T cells through another signaling pathway, or the cancer cells can start to change their surrounding environment to make it suppressive to the immune response. And then finally, CAR T cells themselves can be exhausted. So there are a lot of efforts to address these different problems. And if you look in clinicaltrials.gov, there are thousands of studies that are going on for immune checkpoint inhibitors and thousands of studies going on for CAR T cells. So there's really this huge unmet need where we need to understand, what can we do to T cells to make them better in terms of cancer immunotherapy. And so the big focus of this challenge is really thinking about, what are genetic changes we can make in T cells to make them better at killing cancer? And that's really what we're going to be focusing on across these different tasks in the challenge. And so, to do this, we really need to, at a basic level, understand everything we can about the pathway that leads from these sort of naive T cells to these progenitor, effector, and terminal exhausted T cell states. So we really need to know the gene expression programs and the changes responsible for all these steps. And doing this really has been the work of decades. And furthermore, we would like to not just understand, the gene expression programs that are happening here, but, what perturbations we could make and how they would affect this process. So this is a really slow process. It involves a lot of biological experimentation. However, in the last decade or so, our technologies and our computational approaches have really empowered us to believe that we can make this discovery process a lot faster. And that's what this challenge is really about, of understanding this process of T cell differentiation when they're in these exhausted states, and how we can perturb the T cells to shift them away from those terminal exhausted states. So that's the overview of the biology and the introduction to biology for this data science challenge. And then next, we'll talk about how we actually measure the cells in the immune system, and how we look at their gene expression values, and how we can actually perturb the immune cells to get, maybe more desirable states that would be more effective in cancer immunotherapy.